Okay, so now we talked about the sugars and how they were going to be activated into UDP or, in a rare case, CMP. And now we're going to talk about what happens with these sugars in the case of proteins. So we're going to talk about glycosylation of proteins. So here we're going to have to talk a little bit about what do we mean by a glycoconjugate and how important they are. We're going to talk about how you actually make glycoproteins, some of the differences between glycoproteins and proteoglycans, and we'll talk more about proteoglycans when we talk about extracellular matrix. And then we'll talk about targeting of proteins to the lysosome. There's a lot known about the lysosome, but we also have a lot of diseases associated with a defect in targeting proteins to the lysosome. So a glycoconjugate is essentially an active molecule, biologically active molecule. And so they're going to be very important as energy storage, structure. They're also going to carry information. They're going to allow communication between cells as well as extracellular environment. They're going to label proteins for transport to specific organelles. We'll talk about that with respect to our lysosomes. They're going to be recognition sites for extracellular signals, but they can also be used as targets for pathogens to gain access to our cells. Now, an important thing is they're going to form the glycocalyx, which is basically a carbohydrate layer several nanometers thick that coat our cells. And these are going to be made of sugars, oligosaccharides. They're going to be cell recognition and adhesion within this glycocalyx, migration during development, involved in blood clotting, immune responses, and wound healing. And there are basically three things that can be glycosylated. And you have our proteoglycans, we have our glycoproteins, and we have our glycolipids. So when we talk about glycosylation, we're talking about adding sugar to one of these three things. Now, glycoproteins are going to have chains of sugars called glycans, and you're going to add those carbohydrates in a post-translational modification, meaning that after or while the protein is being translated, you're going to have addition of sugars. Now you're going to have your carbohydrate chains that can be simple or they can be branched. If they're going to be synthesized in the endoplasmic reticulum, this is going to be attached to an asparagine residue. And we're going to use do-glycophosphate in the ER to transfer these sugars to the asparagine residues. Glycoproteins, again, are going to be very important on the cell surface, and it's going to be found in pretty much every tissue. Now, about 50% of all proteins are going to have glycosylation associated with them. We're going to have glycosyltransferases that are going to be important. There are many families of glycotransferases. Again, they're going to be specific for that sugar and the protein modification. So it's a combination. Remember, all enzymes need to bring substrates together. That's their purpose. Glycosyltransferases are going to be important in the synthesis of our glycosaminoglycans, or GAGs. And they're also going to be important for modification in our proteoglycans. So for glycoproteins, here's our sugars that we're going to add. We're going to add glucose, galactose, mannose, fructose, and then of course we have the acetylated N-acetyl glucosamine, N-acetyl galactosamine, unacetyl aminic acid or sialic acid, and all of these are going to increase negative charges on your proteins to allow them to interact with their aqueous environment, the external environment. Primarily, they're going to be on the outer leaflet of your membranes. Sialic acid is going to be so important that even cancer cells will increase its expression to allow them to enter the bloodstream and metastasize. So, pretty simple distinction between proteoglycans and glycoproteins is that proteoglycans contain sugar acids, gluconic acid or iduronic acid, and glycosaminoglycans are building blocks of these proteoglycans. In general, glycoproteins have less extensive modifications than your proteoglycans have. All right, so in the generation of glycoproteins, there's a few different steps that can be taken. We'll talk about some of these. We have N-glycosylation, which is going to occur in the ER. O-glycosylation it's going to occur mostly in the Golgi. O-menoxylation is going to occur in the ER as well. C-menoxylation can occur in the cytosol. And we have glycophosphatidylinositol or GPI linkages that can also occur. And so just remember what we're talking about for N, we're binding for O linked on oxygen. For N linked, of course, it's going to be a nitrogen. Now for glycoproteins, of course, this is all post-translation, so the protein is going to get synthesized on the endoplasmic reticulum, as all proteins do, and then the carb chains are going to be attached either in the ER lumen or in the Golgi. In some cases, it can be also in the cytoplasm. The UDP sugars that we talked about are going to be the precursors for 4 of the 7 of the sugar attachments found in our glycoproteins: UDP glucose, UDP galactose, UDP and acetylglucosamine, and acetylgalactosamine. GDP sugars are going to be for mannose and fructose, and we have CMP is going to be for our sialic acid. So N-linked is going to be the predominant carbohydrate modification for mammalian cells. It's going to be on asparagine residues, and the amino acids is going to have a particular sequence. Important just to note that proline cannot be part of this sequence. Proline gets excluded from a lot of things due to its unusual ring structure. There's going to be a lot of proteins that are going to contain at least one occurrence of this sequence for glycosylation. Enzymes, as we know, are glycosyltransferases. And then this is going to occur in the lumen of the endoplasmic reticulum. And again, we're talking about N-linked for the nitrogen. Here is an example. So we have an asparagine residue here, and then we're adding a lot of different sugars to generate this branch for this particular protein. So this protein is now glycosylated. O-linked glycoproteins are going to be where you have sugars again added to the protein polypeptide. The activated sugars are going to be again UDP, GDP in the form of mannose, or CMP in the form of sialic acid. Enzymes are again same glycotransferase superfamily, and the synthesis is going to occur on ribosomes bound to the ER, and then is going to process through primarily the Golgi apparatus for the glycosylation. So the protein's made in the ER, but the glycosylation occurs in the Golgi for O-linked. O-menoxylation, don't get confused with O-linked glycoproteins. These are different. So when you're starting with a mannose, this is going to be incorporation of mannose on serine and threonine residues. This is going to occur in the ER, and it's going to use glycophosphate as well, similar to N-linked. Oh, I spelled that wrong. Do-lico. So it's similar to the N-linked process. O-menoxylation is going to be on many different proteins, such as alpha-dystroglycan. We're going to have other glycopeptides from brain tissue that are going to get O-menoxylated. Activation of mannose is going to be through the GDP, so that's going to have the energy. It's going to transfer that to do-glycophosphate, and then it's going to build on the sugar depending on the protein. Alpha-dystroglycan is the best studied O-menoxylated protein. This is going to be part of your dystrophin glycoprotein complex in your muscles. And if you have defects with O-menoxylation, it can lead to reduced function of alpha-dystroglycan. And so this is going to be related to several autosomal recessive muscular dystrophies. And these are congenital muscular dystrophies, which can have diverse phenotypes. We have the Walker-Wahlberg syndrome, which the individual will die within the first year. And then there's the muscle-eye-brain disease. And this is going to be less severe. All right, so what proteins get modified? Most proteins that are secreted will have sugars bound to them. Remember, we have an aqueous environment, and the sugars allow the proteins to interact with that environment much easier. You're going to have the extracellular portion of proteins modified in the outer leaflet of the plasma membrane. And you're also going to have it linked to intracellular, either cytosolic or nuclear proteins. And typically these are going to be O linkages, important to the function of the proteins. So we're going to have N-acetylglucosamine, which is going to attach to serine and threonine residues. We have transferases that are going to be specific for these, important in sentinel transduction proteins. We talked a little bit about that with respect to our iris, for example, our insulin signal transduction pathways. So almost all transcription factors are going to be O-glycosylated, and RNA polymerase II is also glycosylated. So this is just an example. So we have you have our protein, which is made in the ER. You're going to go through the Golgi complex. Some are going to enter a secretory vesicle, depending on what they're tagged with. And these are going to either end up being secreted completely, or it can end up on the surface. And the carbohydrates are going to be on the proteins interacting with the aqueous environment. Others are going to be tagged to go to some cellular location, such as the lysosome. And these enzymes will be delivered to the lysosome based on their mannose. So this is O-mannosylation, and they have a mannose receptor that's going to be important for picking those up. So lysosomes are going to be important because they contain digestive enzymes. So the enzymes, of course, are going to be made in the endoplasmic reticulum for messenger RNA, and then they have to get tagged to go to the lysosomes. They're going to digest cells, components that are worn out, such as organelles. They're also going to break down different products. They can also take in viruses and bacteria and break them down as well. And it's important to get the enzymes there. Now you're going to have O-ling. So your glycosylation is going to be added to your carbon-6 of mannose residues that have been added. And then you're going to have your N-acetylglucosamine, 1-phosphate, added to the carbon-6. This is going to be activated by UDP and transferred by the N-acetylglucosamine phosphotransferase. Now, N-acetylglucosamine is going to be removed, and your mannose residues are going to be phosphorylated at position 6 and generate a mannose 6-phosphate. The mannose 6-phosphate is what's going to get recognized by the lysosome. So there's a mannose 6-phosphate receptor on the membranes of the Golgi, and this is going to target the enzymes to the lysosomes. Eye cell disease is a disease in which you lack the mannose phosphate marker, and so you're not going to get your lysosomal enzymes trafficked to the proper place in the lysosome. And so in this case, the phosphotransferase is going to, which typically is going to recognize the lysosomal proteins by their 3D structure and tag them to the lysosome, is defective. So this phosphotransferase is going to be defective, and you don't get the mannose 6-phosphate formed, which is shown here. So your lysosomal enzymes will be secreted from the cell, and your lysosomes will accumulate molecules that they should be breaking down, and these accumulations are referred to as inclusion bodies, and that's where this disease name comes from, the eye cell or inclusion cell disease. And then you also have glycosylphosphatidylinositol, and these are GPI linkages for proteins that are going to get anchored. So you have your glycoproteins tethered to the outer leaflet by the GPI, and glycoproteins are glycated and this is going to be important for our red blood cells, decay accelerating factor DAF. DAF is going to be important on the red blood cell membrane because it helps prevent lysis by complement, which is an enzyme that is going to be found in inflammatory environments. Also important GPI linkages are acetylcholine esterase, and we have some adhesion molecules, neuronal cell adhesion molecules that also have GPI linkages. Okay, so in general, O-linked carbohydrates are added sequentially, and linked, you're going to have branch carb chains, and this is going to be on do-glycophosphate, and it's going to start with asparagine. O-methylation also uses do-glycophosphate.